Hello everyone! Welcome back to G-nius Review Center. Now this video will revolve around DNA replication, one of the most fundamental processes of all living organisms. I am Albert Don Marave, and in this lesson, we'll break down how cells copy their DNA perfectly every time they divide. Let's now begin! Now by the end of the lesson, you should be able to first define DNA replication and explain its biological purpose in cell division and genetic inheritance. Second, identify and describe the role and mechanism of key enzymes involved in DNA replication. Third, illustrate the coordination of enzymes during DNA replication at the replication fork, while also distinguishing the leading and lagging strands. Fourth, analyze how errors during replication occur and how these are minimized by enzymes. Lastly, we must critically evaluate biotechnological applications of DNA replication. Now, before we dive deeper into DNA replication, let's first take a quick moment to reinforce what we've learned so far. This is a fast and fun review activity I like to call building the DNA ladder. Now, the mechanics of this activity is simple. I will be presenting on screen a single DNA strand, and your task is to fill the missing side of the DNA chain. This review is to test your knowledge on base pairing and strand orientation. As I present the single DNA strand, feel free to pause the video and try it out yourself. Are you ready? Then let's begin. Here is our first single strand DNA. If you answered 3' TAG CTA GCC 5', then you are correct. Here is our second single-strand DNA. If you answered 3' GCA TTGCA 5', then you are correct. That's all for our review. Hopefully you learned a lot, and hopefully you had fun with this initial review. Now that we've exercised our brains a bit, here's a question I'd like to ask. Have you ever wondered how your cells make exact copies of your DNA? Every time your body heals a cut or grows, your cells divide, and every new cell must be a complete identical copy of your DNA. That is where DNA comes in. DNA replication is a fundamental biological process that ensures every new cell receives an exact copy of the organism's genetic material. Whether your body is growing, healing a wound, or replacing old cells, replication is what keeps the genetic material consistent, like copying a master blueprint for every construction site. Now, this process is vital not only in the growth of multicellular organisms like us, but also in tissue repair and even in unicellular organisms where it allows reproduction. Without accurate replication, the cells would lose track of the instructions needed to survive and function accurately and properly. It maintains genetic continuity from one generation of cells to the next, ensuring that all cells have the information to function properly. DNA replication is described as a semi-conservative process. This means that each of the two resulting DNA molecules retains one original or parental strand and one newly synthesized strand. Imagine it like a zipper. When you unzip it, you get two sides. DNA works similarly. It unzips into two strands. Then, each original strand serves as a template for building a new strand. So in the end, you're left with two zippers, each made up of one old half and one newly made half. Moving on, let's take a closer look at the replication fork, the central hub where all the action begins. The replication fork is a Y-shaped structure that forms when the double-stranded DNA is unzipped. Now here's something interesting. At the replication fork, the two strands of DNA are not copied in the same way. One strand, called the leading strand, is synthesized smoothly and continuously in the same direction that the fork is opening. However, one strand is called the lagging strand and is made in short, disconnected fragments called Okazaki fragments because it runs in the opposite direction. Now you may be wondering, What are the key enzymes that makes all of these happen? Now before we continue to that portion, let's have a quick review on what we've learned. Please pause this video after the question and try to answer it yourself. Which of the following best explains why DNA replication must occur before cell division? If your answer is B, then you are correct. It is so that we can ensure that each of the new cell receives a complete and identical set of genetic instructions. Let us now meet the key enzymes working at the fork, the ones that form what we call the replisome complex. First up, we have our unzipper. Helicase, our unzipper, is responsible for unwinding the DNA double helix at the replication fork. It breaks the hydrogen bonds between base pairs, separating the two strands so that each can be used as a template. Think of Helicase as the person unzipping a jacket, except this zipper splits the DNA strands. So we also have some associated proteins, for example, the single-strand binding proteins. These proteins immediately bind to the exposed single-stranded DNA to prevent reaniline and degradation. Next up, we have our builder, the DNA polymerase. DNA polymerase synthesizes new DNA strands by adding nucleotides that match the template strand. But they can only begin synthesis at a 3' OH group, which is provided by the primase. Now, the primase is a protein that synthesizes short RNA primers needed to initiate DNA synthesis. These primers provide the 3' OH group required by the DNA polymerases, and it is especially active on the lagging strand, where new primers are needed for each Okazaki fragment. Now, the problem comes with DNA strands being antiparallel, meaning one strand runs from 5' to 3' and the other runs from 3' to 5'. But the DNA polymerase can only add nucleotides in the 5' to 3' direction. This creates what we call the leading strand and the lagging strand, where the leading strand is oriented from 3' to 5', so the DNA polymerase can follow the fork smoothly and continuously. However, in the lagging strand, it is oriented from 5' to 3' already, so the DNA polymerase has to work backwards away from the fork. This is what creates the short fragments called Okazaki fragments. Now, an analogy we can relate this to is that the DNA polymerase is like a construction worker laying bricks, but it can only start if someone else lays the foundation, which is the primase. On the leading strand, it's like paving a straight highway, but on the lagging strand, it's like building a road out of small concrete slabs where you have to put each piece individually and you have to stop frequently to restart. Lastly, we have our sealer, the DNA ligase. DNA ligase is responsible for joining the Okazaki fragments on the lagging strand by forming what we call phosphodiester bonds between the 3' OH end of one nucleotide and the 5' phosphate end of the next. Without the ligase, these fragments would remain disconnected, making the DNA molecule unstable and unfit for proper cell function. An analogy to go by this is that the DNA ligase is like a glue gun or a sealer which seals the gaps in between bricks on the wall, finalizing the construction so that the structure is strong, stable, and complete. Now, to compile what we've learned so far, because I'm sure it's quite a lot of information, displayed now is a table which summarizes all the information that we have discussed. Take some time or take a screenshot of this so that it may help you as you review for the topic. DNA replication is an enormous task. Imagine copying 3 billion letters, which is the number of base pairs in the human genome, every single time a cell divides. You'd expect a lot of mistakes, right? But surprisingly, DNA replication is remarkably accurate, with fewer than one error per billion base pairs copied. How is that possible, you may ask? It's all thanks to the built-in feature of DNA polymerase called proofreading. As DNA polymerase builds the new strand, it does not just blindly add one base after another. It also checks each base as it goes. If the enzyme accidentally adds the wrong nucleotide, for example inserting a T base where the C should go, the shape of the DNA will be slightly distorted. DNA polymerase can then detect this misfit, and once it spots the error, it uses its 3' to 5' exonuclease activity, which is essentially a built-in eraser, to remove the incorrect nucleotide. Then it resumes synthesis by inserting the correct base. Now let's take a quick break to test what you've learned so far about the key enzymes involved in DNA replication. Again, feel free to pause the video and try to answer these few questions. Why does DNA polymerase synthesize the lagging strand in short fragments instead of continuously? The correct answer is C. DNA polymerase can only synthesize in the 5' to 3' direction. Next question. Which enzyme is responsible for sealing the gaps between Okazaki fragments on the lagging strand, forming continuous DNA strand? The correct answer is C. DNA ligase, or sealer. Now that we've explored the key enzymes and mechanisms behind DNA replication in cells, let's take a step into the real world, or rather, into the lab. DNA replication doesn't just happen in living organisms. Scientists have learned to mimic this process in the laboratory using a technique called the polymerase chain reaction, or PCR. PCR is a powerful method that allows us to replicate specific DNA segments millions or even billions of times in just a few hours. This makes it extremely useful for research, diagnostics, crime scene analysis, and more. How does it work? PCR copies DNA using a thermal cycling process, unlike natural replication where it happens inside cells at body temperature. Here are the 3 basic steps of the PCR cycle. The first step is denaturation, wherein the DNA is heated so that the double strands separate, similar to what Helicase does in cells. The second step is annealing. A short synthetic primer binds to the specific target sequences. These are what replace the functions of primase. And lastly, extension. A specific DNA polymerase called TAQ polymerase synthesizes new DNA strands, just like the role of DNA polymerase. So why is this so important to learn? Understanding PCR gives us a window into how we can use molecular biology to solve real-life problems, from diagnosing diseases and solving crimes to advancing genetic research and biotechnology. It also reinforces your understanding of natural DNA replication, because all in all, PCR is simply a stripped-down, lab-controlled version of it. And there you have it, folks. We've just about covered everything that there is to know about DNA replication. We've met the key players, Helicase or unzipper, DNA polymerase or builder, and ligase or sealer, who all work together to ensure genetic information is copied faithfully every single time a cell divides. And we've seen how this cellular process has been transformed into a powerful laboratory tool in the form of PCR, which now plays a crucial role in medicine, forensic science, and even genetic research. Before we conclude today's discussion, I've prepared a short interactive activity to help you review and apply what you've learned today. Just scan the QR code on the screen to access a quick challenge. It will only take a few minutes and will help reinform the concepts that we've learned today. Use this as a moment to test your understanding and maybe even discover what part of the process you like the most. That's all for today's lesson, everyone. Great work, and I'll see you in the next session.